It's a real pleasure to have Dr. Archishman Raju with us today. So Archishman did his PhD from Cornell, thinking about some very interesting problems on how to coarse grain and think about effective models and connected them to information theory and so on. And then from there, he moved to Rockefeller Institute, where he then started working on biological problems, in particular developmental biology, in collaboration with Eric Sigia and Stan Leiber, two of the stalwarts in the field of biophysics. So today he'll talk about some of the things and what he has been doing at NCBS. So yeah, you moved to NCBS 2 years back, right?
Yeah.
So he joined NCBS and of course has been doing some really cool work. And by the way, there is also another aspect to Chishman's work. He also thinks deeply about history and philosophy of science, and he keeps writing articles, and they get published in The Wire and other places. So let's welcome him. And
Okay, thanks Sandeep, and it's very nice to be here, and I've had some great conversations throughout the day. Today I'll be talking about, as the title says, geometric models of self-weight specification, and this work is done with the people mentioned on the slide. So let me get into it. So just to give a brief outline, I'm going to start with, should I reduce the, okay. So I'm going to start with just saying a little bit about development and developmental biology, and mainly what I need. I'm going to talk a little bit about systems biology models, and then I'm going to try and introduce a framework that was really used by early experimental embryologists, in particular Waddington, to think about development as a dynamical system. Then I'm going to talk about how we think we can, at least for certain, in low dimensions, we can classify the qualitative possibilities of these dynamical systems, and how I think that's useful to fit biological data. I'll give you a particular example using embryonic development in the mouse. And finally, in the last part of my talk, I'll talk about spatial patterns and how to think about some of these spatial patterns in a geometric manner. Okay, so to give you a sense of what kinds of or how you would think about cell fate in development, there's this cartoon where you start with a single cell and then these single cell divides, and then at some point differentiates. Different cells of your body end up taking a different fate. You can have a skin cell, you can have a neuron, and so on. And so you could imagine that at an early stage in embryonic development, you put two different marks and two different points of the embryo, and then you just see what they look like after the organism is developed for some time. And early embryologists used to draw these cell fate maps where you could really keep track of how these cells were and what fate they were taking at the end. And this was sort of seen most starkly in C. elegans, the worm, where you can really keep track of what every cell is doing. So of course in more complex organisms, the cells move around and stuff. When the C. elegans has about 1,000 cells, you can really see as they develop and as these cells divide, what fate does each individual cell end up taking, and it's a very stereotyped trajectory. So the important thing to keep in mind is that these cells form these discrete types, and there's not a lot more that they can do. There are a few discrete types of cells, and in development there's a certain amount of robustness in what kind of cell type you end up becoming, and so in various different organisms you see a trajectory of embryonic development that looks similar, roughly speaking. And the reason that this is a bit of a mystery is if you think about what's happening at the level of the single cell. And so here's a diagram of what's happening inside a single cell, where there's some signal from outside, some protein, for example, which protein is telling the cell what decision to take. And here's an example network of the kinds of interactions that go inside the cell cell. There are various forms of proteins inside the cell that form a complex regulatory network. This gene regulatory, and this regulatory network, or in this case, this is a signal transduction network. You take an external signal and you take it to the nucleus. So these regulatory networks essentially decide how the cell ends up taking the fate it does. And the reason I say it's a bit of a mystery is because if you think about modeling the network in particular, right, and you imagine that all of these components which are important for a particular decision the cell has to take, then you could imagine taking each one of these components and writing down a differential equation for each of them, right? And so, you can do this. And so on the right hand side, right, these have been deliberately drawn too small to see to show you the kind of complexity that kind of a model would involve. And you can zoom in on one of these equations and you see right, that these are typically equations from chemistry. So to begin with, these are already phenomenological equations, either Michaelis-Menten kinds of equations or mass action or Hill's functions. There are various ways to try and model what's happening in the cell. And each one of these equations have a bunch of parameters. There's some rate constants, there's some dissociation constants, and in most cases, none of these parameters are very well measured. And so you could imagine making a model, a dynamical systems model of what's happening inside the cell. And at this point, these models, because of the amount of information we know, the amount of components that are involved in these decisions that the cell takes, these models have a bunch of unmeasured parameters. So in a sense, from the perspective of modeling this system, the question is, how do you get this simple behavior where you have a few discrete cell types in the cell from something that's seemingly so complicated? And the interesting thing is that early experimental embryologists used to think about the way the cell is taking a decision in a different way. And in particular, Waddington, who was doing these experiments in embryology, he had a very different idea of how the cell is taking a decision. So instead of looking at the molecular components involved in the cell deciding to take a particular kind of fate versus a different kind of fate, Waddington drew this kind of landscape picture that's become quite popular recently, where you have a ball rolling down a hill. And you see that there are these different valleys, and these different endpoints, he said, correspond to different fates that the cell can take. And what he was trying to capture with this landscape picture is to say precisely this idea that actually the dynamical system inherently in development, the dynamical system involved is very robust, and the trajectories you take sort of take you along these predestined directions, and there are only a few endpoints that you can reach. The picture was meant as a metaphor, but it's not something I'm going to talk about. But in fact, his idea, his view of what's happening in development came from a somewhat philosophical difference with a more molecular picture of what's going on. And in some ways, things that became popular in physics a little later, ideas of emergent descriptions, was something that I would argue were, similar ideas were being explored in biology in the 1940s and 50s. So in any case, what this allows you to do, what this Waddington metaphor allows you to do, is to think about development as a dynamical system, which is not very high dimensional. So if you think about everything that's going on inside the cell, you can imagine making a list of all the components involved in the kind of diagram I showed you earlier, the various genes and so on. And these things now can even be measured. You can really sequence sort of the gene activity in a single cell. You can measure all of the gene's activity. And you can imagine making a list of differential equations, right, where the left-hand side tells you how the activity of the gene is changing over time, and the right-hand side is, something that is given by the chemistry, that's given by however these things are interacting with each other. And the kind of main insight that Waddington had was that even though this is a large dimensional system, effectively there is a lower dimensional manifold in which this dynamics is taking place. And in this lower dimensional manifold, you have some standard concepts from dynamical systems which correspond to biological concepts. So for example, what we would call a stable fixed point is, right, in the Waddington picture, this would be a fully differentiated cell. And of course, what's most important from the dynamical systems perspective is that you can have these different stable fixed points that are arranged in some way, and you can decide between these two, between these different you can decide between these different fixed points at the saddle. And so the saddle points, in a sense of the dynamical system, classify how the cell ends up deciding where it wants to be. The kind of approach that I'm going to talk to you about today is to ask if we can start from the opposite end. Instead of trying to look at all of the molecular details that are involved in a cell taking a decision, can we start classifying the possibilities for low-dimensional dynamical systems? Given that we know that many developmental decisions are intrinsically low-dimensional, can we thus make effective models of what's happening in development, and can we classify some qualitative possibilities for what these dynamical systems could do? And there's some old work which we're building on by René Thom in particular, which says that Waddington landscapes, this idea that you have these robust developmental trajectories, are linked to what are called Morse-Smale systems. And the main thing in Morse-Smale systems that links to ideas of Waddington is this concept of structural stability. And structural stability, very loosely speaking, is the idea that small perturbations don't change the qualitative nature of the dynamics. Your dynamical system remains qualitatively the same if you make sort of arbitrary small perturbations to it. And this is of course linked to the biological idea of robustness, which says that if a biological system is very fine-tuned, then it's something that's sort of intrinsically not robust, and small perturbations will change the behavior. There's a theorem by Smale in 1961 which says that if you take a vector field which is structurally stable, and there are certain other conditions you have to impose on it, which I won't go into, they're not going to be so important for the talk, but basically you want to disqualify things like chaos, and in fact I'm not even going to talk about periodic orbits. But essentially, if you have a dynamical system which has a finite number of saddle points and attractors, then you can always write that dynamical system as a potential. So you can always write the dynamics as a gradient of a potential, with the caveat that you need a metric. You give me any vector field, it has to satisfy a certain number of conditions, it has to be structurally stable. And remember, structural stability we are justifying in development from the idea of developmental robustness. Then you can always write that dynamical system as the gradient of a potential times a metric. And what this theorem does is it really formalizes the Waddington metaphor. It says that when you're drawing a landscape for a cell going downhill, you can in fact come up with these gradient flows which tell you how an arbitrary dynamical system is taking these decisions, but with the caveat that you need a Riemannian metric alongside that gradient flow. Okay, so to give you an example, right, a very trivial sort of example, you can imagine having a bistable system, and the flow diagram for the bistable system, which is just derived from a potential, would be something like this. You have a saddle point in between, you have two stable fixed points, but what you can do with the metric is you can change the flows. So you can change the flows. For example, in C here, I'm showing something that is derived from exactly the same gradient flow, but I've added a Riemannian metric. And so the different information that the potential and the metric has is that the potential is always going to have the fixed points, it's going to have all of the saddles and the stable points. The metric is something that can modify the dynamics between them. So if you see these two plots, the stable fixed points and the saddle point in between are exactly the same place, but the dynamics of how you go from here to your stable fixed point is very different from in this to this figure, even though they derive from the same potential. So the metric is an extra piece of information that you need in these kinds of gradient flows because they really tell you where you're going to end up. And of course, this is essential from the developmental biology perspective because we want to think about these different stable fixed points as sort of different kinds of differentiated cell types. Again, this could be at the relevant time scale, so they don't need to be really the final cell types, but at a relevant time scale, you want to think about these things as differentiated. If you do that, you really want to know sort of how it gets there from the decision making point. So what this theorem of Smale does for us is that it tells us that in a typical developmental system, assuming that given some mathematical assumptions which are biologically plausible, you should always be able to write that dynamical system as a potential flow if you include a Riemannian metric. Okay, but what you're interested in doing in biology is not just having your potential, not just saying what will happen to a particular cell, where it will go. You're interested in parameterizing the landscape. You're interested in making mutations. You're interested in putting in proteins. You're interested in sort of perturbing the system and seeing where it ends up. And so you have a theorem which says that you have a potential. Now you want to parameterize that potential and say where can I end up given different kinds of biological perturbations that I might want to do to it. So it turns out that to right now think about how you can make qualitative changes to the systems, right? So you have these structurally stable systems, but you also want to be able to qualitatively change them because you want to be able to externally perturb them biologically, and you want to see what that does, and you want to be able to capture that in your model. So to do that, it turns out that you just need two types of bifurcations, and to argue that there are only two types of bifurcations that you need to talk about how different landscapes are connected to each other, you again impose this condition of genericity. It turns out that, of course, we know about many different kinds of bifurcations, but all complex bifurcations sort of require certain special symmetries. And there are only two kinds of, in a sense, simple bifurcations, generic bifurcations, which are the saddle node and something called the heteroclinic flip. So the saddle node is familiar, right? You have, let's say, three stable fixed points in two saddles. The saddle and the stable fixed point can collide, giving you a saddle node bifurcation. The heteroclinic flip is a global bifurcation. So what it says is that it changes the connections between different fixed points. So in this picture, what the heteroclinic flip would do is it would shift the positions of these two saddles relative to each other. And you can, of course, shift them to the right or you can shift them to the left. And why the heteroclinic flip is important for us is that the heteroclinic flip really tells you if you start from this state, if you exit this attractor, let's say if you're driven out of this attractor by noise, you want to know where you end up. Do you end up here or do you end up here? So the heteroclinic flip is a global bifurcation which is not really changing the nature of the fixed points, but what it's doing is changing their connections. And these connections are essential from a biological perspective because you want to know if I leave a certain type of cell, as my cell is differentiating, if I leave a certain state, where do I end up? Okay, so yeah.
What has stochasticity? You said you have noise, but I didn't see any noise.
You can add noise, yeah. I mean, I'm not going to discuss the noise so much, but you can sort of, add Langevin noise or whatever you want to add to these equations.
Because the main dynamical system, the equation of motion you started with, right?
Yeah.
I have not heard any noise.
That's right. Yeah. So I guess so. Right. I mean, so there's a separate question you can ask: are the kinds of noise you can put in this system also universal in some way? Are there specific kinds of noise terms you can put in? I mean, so far in the general way in which biological noise is treated, where it's some sort of black box which you put in some Langevin term, we have sort of been treating noise in that sense that you first have your potential and then you add some, you add some Langevin term for the
noise. You always, return to the deterministically, all these fixed points.
Yeah, sure.
Whatever you do doesn't matter. You always fix there.
No, you're always going to have noise, but I'm not going to say anything very particular about the noise, except to say that you can have your dynamical system, you can add noise to it. Yeah. Okay, so again, to get back, so we have our landscape, we have different kinds of attractors and saddle points, and we want to see what kinds of bifurcations they take. How can you qualitatively change the nature of the landscape? And right, you can imagine getting these bifurcations as you change a particular parameter. And now if you imagine a 2-parameter space, right, you have some 2 parameters which control your landscape, you can imagine that there's a curve of bifurcations in this 2-parameter space. So in fact, this curve I got from the potential which created this landscape. You know, there are 2 parameters in my potential, and I changed those parameters. And right, you have this curve here, and as you transition from this part to this part, you get a saddle node bifurcation of this upper fixed point. And as you transition across this dashed line, you have a heteroclinic flip. So you have a global change in the connections of the different fixed points. And these 2 kinds of bifurcations are going to be sort of enough for us to figure out how in these, if I take a low dimensional landscape, in fact I'm going to restrict myself to landscapes which have only 3 stable fixed points, how can I transition between different qualitative possibilities. And when I discuss the example, you will sort of see concretely how different qualitative possibilities can sort of be biologically informative. But for now, this is sort of abstract. I'm restricting myself to low dimensions. You could do this for higher dimensions, just the counting gets harder. But imagine if you have 3 stable fixed points, you know, how do you, and you imagine all sorts of generic bifurcations that could occur. You could get these saddle node bifurcations which get rid of this point or this point or this point. You know, can you make a list of all of the qualitative bifurcation diagrams you could get in a 2-parameter space? I'm also restricting myself to 2-parameter spaces because again, it's typical of how many parameters you would control in these developmental biology experiments. You typically can't control everything, but right, there's some 2, for example, signaling proteins that are involved in the decision, and you can control the levels of those proteins. So with 2 parameters and 3 stable fixed points, we can ask, can you make a list of all of the qualitative possibilities? And it turns out you can make arguments for how these lines of bifurcation should meet each other, and you can sort of classify the qualitative possibilities for the dynamics into certain classes, which are names is derived from sort of older things in catastrophe theory, which were more local, but the main point, again, the details of what these landscapes are like are not so important for this talk, but the main point is that you've restricted yourself to a lower dimensional space. The argument for restricting yourself to a lower dimensional space comes from the biology, the fact that you don't get mixtures of cell types. You really get these discrete cell types when you do biological perturbations. When you restrict yourself to a low dimensional space, you make the assumption of structural stability, again deriving from the biology, and then you ask what kinds of bifurcation diagrams I can get. It turns out that you really have a few possibilities for the bifurcation diagrams. So again, to sort of reiterate what this means, you can take whatever gene regulatory network model you want. For a system like this, you can take vary 2 parameters in that system, and you're going to produce, as you vary those 2 parameters, you're going to produce one of these diagrams. So in that sense, this is trying to classify the qualitative possibilities that exist for the dynamics. And the reason we think that this is important is because, of course, then this gives you, in a sense, an effective model for what's happening biologically, which you can make without having to refer to the underlying molecular details. Okay, so to make this a little bit more concrete, I'm going to discuss a particular example.
So you're changing the parameters, and that changes the connections. But the fixed point stay fixed, stay where they are.
You could change the location of the fixed point. I mean, that's not a qualitative change if you change the location. You could remove, also with the saddle node, you could remove the fixed point, so that's also allowed. But the location is not something we're attached to. That's qualitatively the same. Okay, so to give you an example of why this would be relevant, I'm going to take early embryonic development in mice. And so in early embryonic development in mice, which are meant to be closest as a model system to humans, you have a single cell. As usual, development starts with a single cell. These cells divide, and then you have 2 binary decisions. The first binary decision is, right, you go from something called blastomers to two different types. I mean, these have names which are not important, trophectoderm and inner cell mass. And then you have a second binary decision where these purple kind of cells divide into red or blue. This is called the primitive endoderm and the epiblast. The epiblast is what ends up becoming the embryo proper eventually. And so I'm going to focus on this particular section of the decision where the inner cell mass ends up becoming either the primitive endoderm or the epiblast. So naturally you can see there are three kinds of states here, and again this is quite typical of developmental decisions where you have something that's differentiating as you go along in development. So these are days here. As you go along in development, you have things that are differentiating, and so for the relevant time scales, of course eventually the epiblast is going to differentiate further, but for the relevant time scales you can assume the dynamics is captured just by having three states there. One is the inner cell mass, the primitive endoderm, and the epiblast. Okay, now remember we talked about how you can have perturbations that change the nature of the landscape. So we want to figure out what this landscape could be, but we also want to figure out how to change it. And again, this is a system that's been very well studied. It's been really, in a sense, from the biological perspective, it's been beaten to death. But it turns out there's something called the FGF signaling pathway, where FGF is just a protein, and as you increase the levels of that protein, you get all primitive endoderm. As you inhibit the levels of that protein, you get all epiblast. So in a typical cell, in sort of the what I call wild type conditions, if you did nothing to the embryo, you would get sort of about half and half of these either primitive endoderm cells or these epiblast cells. But as you vary the levels of this protein, which you can do, you see that the cells don't end up becoming half and half, but they all become primitive endoderm and epiblast. Again, it's important to remember here that as you vary the levels of a protein, nothing crazy happens. The cells still divide, they still differentiate, but they still differentiate into two types that biologists can recognize. And again, this is part of our assumption that as you change a parameter in the system, you are not making, the thing is remaining structurally stable, and you're not making any sort of fundamental changes to the dynamics. You're not getting, for example, new kinds of cell types. Okay, so now, you can again look at what's happening inside the cell, and that's something you can do. And again, turns out this protein, there's some genes involved. People think these genes, mark the expression of either the epiblast or the primitive endoderm. And this is sort of a very simplified circuit of what's going on. So you can make a circuit like this, and you can write down a list of equations for how the FGF protein is deciding for the cell whether it wants to become one type or the other type, and so that's that kind of a model would try to look at the molecular details. But from the geometric perspective, you want to do something different. You want to say what could this landscape be, and are there different possibilities for how these fixed points, the inner cell mass, primitive endoderm, and epiblast, are connected to each other. So to get to the landscape, actually first we had to do something different. We had to realize, we had to talk about how these cells are talking to each other. So far, I've been talking about decisions at the level of a single cell, but of course in embryonic development these cells talk to each other. That there's, cell can secrete a certain protein which another cell can have a receptor for, and then that can change the decisions in that other cell. And so we need some model for how these cells are interacting with each other. And in this case, again, because the blastocyst is a well-studied example, you can actually sort of track the levels, not directly of the FGF protein, but something that's sort of connected to it called the ERK. So you can image the ERK, and then you can ask what are the spatial correlations of the ERK. Are cells just talking to their neighbors? How well correlated are the ERK levels in a particular cell with its neighboring cells? And so this is something we did for the live imaging data. This is sort of incidental, but it's needed to build the model, and it turns out that there's sort of no spatial correlations in the ERK. So what this means is that this protein, protein which, right, I told you is required for the cells to decide whether to become primitive endoderm or epiblast, which is also a protein that the cells can secrete and that the cells can read through a receptor. This protein seems to be sort of not very correlated inside the embryo. So this protein, of course, diffuses, the cells move, and so there's nothing saying that there's sort of any very local interactions in the way the cells are talking to each other. And that's useful for us because it just means that you can assume that this parameter, this external protein which is determining these decisions, is sort of mean field. And once you assume it's mean field, you're able to sort of simplify the model. And then the next question you have to ask is just what kind of geometry will there be. So from the geometric perspective, you don't try to look at the molecular details. What you say is that the dynamics is in some abstract space. This abstract space is some lower dimensional manifold of the large dimensional manifold that's actually determining these decisions, which is the set of all the gene activities. So it's some lower dimensional manifold in this large dimensional space. There are three states that you know phenotypically exist: the inner cell mass, epiblast, and primitive endoderm. You assume that the protein that's determining for a particular cell what decision it should take is sort of mean field. Right? There are no sort of strong interactions between cells that you want to, there are no local interactions between cells that you need to consider. There's just some mean field total level that each cell sees. And finally, another sort of assumption that we had to make was that these epiblast and primitive endoderm are sort of symmetric states, and these are again assumptions that you can change. Okay, but then sort of the fundamental question that you need to ask from the geometric perspective is how these states are connected to each other. So now you have these three states: the inner cell mass, primitive endoderm, and epiblast, and there are basically two sort of possibilities for how they could be connected to each other. So the potentials are drawn here, and these are, right, the corresponding streamlines for these potentials. And one possibility is, right, that the inner cell mass cells exit this attractor, and they can exit in different ways, right? So they could exit it using noise, or they could exit it because there's a saddle node bifurcation. But they exit the ICM attractor, and they decide to become either the red type, or they decide to become the blue type, so the primitive endoderm or the epiblast. So that's one possibility, that you either go here or you go here. Notice that this geometry is sort of fundamentally one-dimensional. Even though I've drawn it here in 2 dimensions, you can draw a curve that connects all of these points, from the epiblast to the ICM to the primitive endoderm. A different possibility is a geometry that's fundamentally 2-dimensional, where you have a different set of decisions. The inner cell mass doesn't just decide whether to become one type or the other type. It first decides to become something that's not inner cell mass, and then it takes a decision whether to become primitive endoderm and epiblast. And again, these are geometrically distinct possibilities for how the dynamics could proceed. So whatever low dimensional manifold encompasses this dynamics and has these attractors in it and these saddle points in it, right, it has to sort of, the connections of these fixed points, they're sort of, in this case, two distinct possibilities for it. In both of these cases, you're going from the inner cell mass and you're becoming either primitive endoderm or epiblast, but they're connected to each other in different ways. The kind of suggestion we're making is that these kinds of geometric possibilities would be distinguishable by experiments, and what you need to do for to do experiments to really distinguish these different qualitative possibilities for what the landscape is like is to have sort of time-dependent perturbations on this system, right? So remember earlier I had shown you the embryo is developing, the inner cell mass cells are becoming either one or the other type, and if you make sort of time-dependent perturbations in these experiments, it turns out that for these two different geometries, you get very different endpoint results. So that means I can take a pulse of my protein, I can put it at some intermediate time, and then I can ask at the end of the day, how many red and blue types of cells do I get? And depending on how the attractors are connected to each other, I'm going to get very different results. So right. So for one of them, the first one that I showed you, things are not going to depend so much on when I put my pulse. For the other one, they're going to depend very sensitively on when I put my pulse. So there are sort of experiments that you can do that sort of concretely distinguish between these two possibilities. As a corollary, what you also get is that whatever gene regulatory network model that you make for this decision, you can ask what the geometry of that model is. So it turns out, right, for the gene regulatory model that exists, the geometry is sort of more like this like this. And if it turns out that time-dependent perturbations, show results like these, then it would tell you that the gene regulatory model has something missing, and there has to be something else that goes into how these cells are taking the decisions. So in the tradition, I guess, of phenomenology, what this is saying is it's giving you a way to minimally parameterize the decisions that the cells are taking, and also make sort of predictions for what would happen when you make perturbations. And in particular, the most informative perturbations are those that are sort of time dependent, where you don't, again, in biology, sometimes you knock out genes, and these are right once and for all, but you can put in proteins in between the developmental dynamics, and what we found is, again, that these time dependent perturbations will be most informative. And these are not something that's usually done, so it's a suggestion for what kinds of experiments could take place. Okay, I'm going to move on now to thinking more about spatial pattern formation, and right, so far I was talking to you about I was talking to you sort of more at the level of, for a single cell, what kind of landscapes it could have, and then I sort of talked about how cells can communicate with each other in the example of the blastocyst, but this was sort of incidental. I sort of just made a mean field model where these spatial interactions are not so important, and so in this next part of the talk, I want to talk a little bit more about how spatial interactions can also be thought of in terms of geometry. And of course, the sort of paradigm for spatial pattern formation is, especially in developmental biology, is the Turing pattern, and the Turing pattern, which of course was suggested 70 years back, is is something that gives you, so here with the figures I'm showing you, whether for seashells and fishes and so on the left is the actual thing, on the right is a computer simulation. So you can get something that looks very much like patterns that we see in nature, and this seems very appealing, that you can take some activator inhibitor system, which I'll talk about in the next slide, but you can take some system, you can take some reaction diffusion system, and you can get spatial patterns that seem to mimic what's happening in nature. There's sort of an interesting story here, because in fact Waddington was not ever convinced about the efficacy of Turing patterns for development, because he thought that Turing patterns are mostly noise driven, as I'll talk about in a second, which many of you may know, but he said since they're mostly noise driven, there's no way that they could be robust, and so maybe they're important for these kinds of patterns that you see and they look nice, but they couldn't be important for anything important. Another way to say it is, actually, if you think about another classic place where Turing patterns are used is the stripes of the zebra, and in fact Turing himself said, well, the stripes are easy, but what about the horse part? That's the real difficult thing to get. Okay, but what I'm going to talk about here is to think about whether we can look at these spatial pattern forming systems and also look at them geometrically. Can we also build a landscape for these systems? The classic kind of model for these Turing patterns is something called the Girer-Meinhardt model. Again, many of you may know this, but you basically have something called an activator which activates itself, and an inhibitor, and the inhibitor inhibits the production of the activator. You can write down a set of equations. I've actually slightly modified the original Girer-Meinhardt model here. That's not very important, but if you solve it with periodic boundary conditions in one dimension, you get some pattern. So here's the activator in blue, the inhibitor in red. The activator and inhibitor sort of follow each other, but there's a bump. So that's sort of the simplest thing you could do with the Turing pattern, right? There's some spatial bump. It turns out if you try to do, so you have some diffusion, you have some two variables. If you try to do sort of the simplest thing you would want to do to write this as a potential, it turns out to be quite hard to do. And the reason for that is, that you can just look at these cross terms of of the Jacobian, and these cross terms are not equal. And it turns out to be actually very difficult to write down a local potential of two fields where one acts as an activator and one acts as an inhibitor. I won't go into this, you can believe me, I'm happy to discuss this later. But to write down a local potential where one acts as an activator and inhibitor turns out to be very difficult. Mainly it has to do with the fact that these cross terms have different signs. So anyway, so this, to just take these equations and try to write down a potential from which they derive in the normal way is is a very difficult exercise. So we thought about this for some time and realized that actually it's much easier to think about these Turing patterns in Fourier space. And in Fourier space, what's happening in these Turing patterns is that you have a homogeneous state, and in the homogeneous state there's some unstable modes. And again, this is sort of part of a classic exercise if you work out what Turing was doing, that there's a dispersion relation and there are a few modes that are unstable. And so in Fourier space, then you can ask what is, instead of thinking about it in the original space, can I think about the geometry in Fourier space? It turns out the geometry in Fourier space is much simpler to think about, and the simplest example of that is if you have just one unstable mode. So in this case where you're just getting one bump, let's say you have just one unstable mode, and you start off at the homogeneous state, which is an unstable fixed point, and then as you evolve in time, it turns out that the geometry is just given by sort of a circle of fixed points around. And this circle, each point on this circle is a stable fixed point which corresponds to a spatial pattern. So because in Fourier space you just have a single unstable mode, all of the other modes, all of the other directions are sort of attracting. You can just make a two-dimensional space for that single unstable mode. You have a cos and a sine, so you have two dimensions. And in that two-dimensional space, the geometry looks very simple. You just have a circle of fixed points around whatever you decide to be the center. And these circle of fixed points, of course, just reflect the translational symmetry in the system. You have periodic boundary conditions, so the position of the bump can be anywhere. But fundamentally, you're really doing something very simple. You're just going from one unstable fixed point to a stable fixed point. Interestingly, the equations for how you go from the unstable to the stable fixed point, are universal. They're always going to have to lowest order, they're always going to have this term where you'll have some nonlinear flow away from the unstable fixed point, and you will have some cubic saturation term. And so you can take your Turing system, you can project it onto Fourier space, and you can look at so I can take the system that I had and I can look at what, as I project it onto my Fourier components, what the dynamics looks like, and I can fit that dynamics very well to a simple equation like this, and where I just have these two parameters alpha and beta, which are now universal. They don't at all depend on the kinds of equations that I start with. And the reason this is interesting is because in fact one of the problems with the Turing mechanism has been the difficulty of identifying molecular components which give you an activator inhibitor kind of form. So it turns out actually if you play with two components, there are very restrictive possibilities for what will give you Turing patterns. There are only two kinds of connections. Activator inhibitor is the most usual one, and even though there's some biological examples where you can identify the activator inhibitor, in general it's very difficult to figure out what the activator and inhibitor is in a biological system. And so to think about it from this potential viewpoint, you're saying that really you just have some pattern in space that's starting from something that's homogeneous with small fluctuations, and then there's something that gives a bump. Okay, but of course, you might want to add more modes. You don't just want one unstable mode. And it turns out you can add more modes. So here you have something with two modes, and again the equations turn out to be universal. You can just write down the differential equations for how components for different Fourier modes evolve, and those equations, which it's easier to write down in complex space, these equations turn out to be universal. To some extent, I mean, this is something that's not that much of a surprise. It's something we're familiar with from Landau theory. But the only point being made here is that you can use these kinds of the equations now to fit to biological data where you might have a putative Turing system, where you think there's a system there's a Turing mechanism involved, and you can fit to the biological data at the phenotypic level without necessarily having to identify what the molecular components are which are really determining that decision. Okay, so just to see how this does, I've given an example here of a simulation in blue, and then a fit to a potential. And of course, this is not, you can't just look at the Fourier mode. You also need something that captures the shape at the final fixed point, and so you need to tweak things a little bit. But essentially, this is sort of, if you don't know sort of the equations involved, it's sort of the minimal parameterization you would have geometrically of how spatial pattern formation takes place through Turing. And again, to sort of emphasize this, you can imagine imagine something like this. So here what I've taken is I've taken a two-dimensional Turing model, and I've sort of randomized the parameters. I've started with the activator inhibitor, right? I sort of added randomness in the parameters with space, and then right I get some complex system where I get I get these 3 bumps. So this is the original Turing molecular model simulations, and these bumps form at different points of time. So initially you have something, right? Initially you have this bump forming, then at some point you have these 3 bumps. They're also, right, they're not symmetrical, they're placed at different places. And right, what you can, so again this is actually typical, something you might expect biologically, but what you can say is you can take each one of these bumps and you can project it on to the correct space, and you again see that all of the dynamics follows this kind of a universal curve. And you can again compare sort of the this potential version with the full Turing system, and again you do very well for what these the dynamics of these Turing systems. And this is relevant because actually recently there's been some interest in how the formation of digits, which just means your fingers if you like, in mice might have a Turing system involved. So recent work has been, again, because you have difficulty in identifying activator inhibitor pairs, there are people who have been trying to look at these 3-component Turing systems, and then also people who've been trying to look at 5-component Turing systems. And again you can write down a bunch of equations for how which produce some pattern. Again there's some very local mode which is unstable. And to give you a sense of how this compares with experimental data, this is some 3 genes that are being tracked, and this is what you would get from a typical Turing simulation. So this is all from this paper, and the idea is that again you're capturing this data, of course, at sort of a very qualitative level, but you're capturing the fact that you get a finite number of digits which corresponds to a finite number of peaks. So the claim would be, again, these 3 components you think are important. There may be other components that are important, but you could take something like this and you should be able to fit it to exactly the kind of potential model that I was discussing with you, and it turns out, again, you can do this. You again get this sort of universal equation. Again, this is all in the spirit of Landau theory, for how your spatial pattern evolves from homogeneous unstable state to a final stable pattern, and what it also shows you is that all of these components all are sort of connected to each other. There's just one unstable eigen direction which is going from the homogeneous state to the final steady state, and all of this can sort of again be minimally parameterized by sort of a simple set of equations in Fourier space. Okay, so Turing mechanism then is an example of something which is intrinsically low dimensional. You can start off with a system that, because the space involved, in principle you have a very high dimensional system, but the dynamics of the system is confined to low dimensions, and what I've argued for you is that in the spirit of Landau theory you can capture that dynamics with a simple potential model, and that you should also be able to use these kinds of potential models to fit data in systems where you think the Turing mechanism is involved. But it turns out that it's not necessary for you to always get these low dimensional systems, and there are cases where you can get more high dimensional dynamics. So again, there's sort of a recent model where this is about bristles and the fly, where you have 2 kinds of fates. You can sort of ignore all of the details on the slides. The main point is that there are 2 points of fates. There are 2 kinds of fates that your cells can take. One is a bristle, which is something, at this point it's called a sensory organ precursor, and the other the epidermis. The other is just your skin, if you like. So either you get bristle or you get skin, and what you see as you image these flies, the back of these flies, is that you get these small dots which are the bristles, and mostly you get skin. And so the question is, the formation of these bristles, which is also a spatial pattern, can you also capture what kind of model would capture this kind of a spatial pattern, which is now not in the form of your usual Turing mechanism. And these people made sort of a clever model for this, where they said that instead of trying to look at again, let's look at it phenotypically, and let's say our cell can again two fates. You can either be epidermis or you can be a sensory organ precursor.